Auf den Spuren der Schleimpilze. Ein Podcast für Neugierige. Hello and welcome to the fifth and final episode of On the Trail of Slime Molds. My name is Anna Bursac. As part of my final research project for high school, I created this podcast series to explore the fascinating world of slime molds. In previous episodes, we've discussed many fundamental characteristics of slime molds, but today we'll take a closer look at one particularly intriguing topic. How do slime molds really move? If you've ever come across a slime mold in the forest, you may have experienced the following. You find it and carefully document its location, only to return a few days later and realize it's gone. Most likely, it has moved. Perhaps because it found a place more to its liking. But how exactly does movement of Myxomycetes work? That's a question we haven't explored yet. When I first learned that Myxomycetes can actually move, I was amazed. How can a single-celled, slimy organism possibly be mobile? How can a slimy, single-celled organism possibly be mobile? For animals like us, humans, the answer seems obvious. As vertebrates, we use our limbs to move. Arms and legs, wings or fins. But a slime mold? Arms and legs, wings or fins. But a slime mold? What is actually going on here? What is actually happening here? To answer that question, let's take a look at the example of Physarum polycephalum. Imagine its plasmodium as a fan-shaped structure spreading outward across the surface. Its broad front slowly probes the environment and advances forward. Always remember, though, this organism consists of just a single cell. Inside that cell, there are the cytoplasm and countless cellular components, the living substance of the organism. All of this material, along with dissolved nutrients, is continuously transported within the plasmodium. This transport occurs through a process known as peristaltic pumping. But how does that work? Through physical signals. Yes, you heard that correctly. Physics plays a central role here. The veins of the plasmodium receive mechanical signals generated by the movement of the cell fluid. These signals essentially tell the veins, expand, contract, expand, contract. Sound familiar? Our muscles operate in much the same way. They contract and relax again. Remarkably, slime molds use the very same protein filaments that make muscle contraction possible in animals. These proteins are called actin and myosin. They are embedded in the walls of the plasmodial veins, and they are also found in the walls of our own blood vessels. These protein filaments contract and relax rhythmically, and the cycle repeats approximately every 100 seconds. Scientists refer to this process as a periodic pressure oscillation. As a result, the direction of the cytoplasmic flow within the veins changes repeatedly. This is why time-lapse recordings often make the plasmodium appear to pulse. The activity of actin and myosin also causes calcium concentrations and ATP levels to oscillate back and forth throughout the organism. So within a plasmodium, we find a network of vein-like tubes that behave in some ways similarly to blood vessels. Inside these tubes flows cellular fluid carrying nutrients. The tubes or veins branch repeatedly, creating a complex transport network. In fact, the plasmodial network of Physarum polycephalum has often been compared to the human circulatory system. And this comparison is precisely what makes slime molds so interesting to medical researchers. One area of research in this field focuses on understanding how changes in these vein networks arise. Scientists do not yet have all the answers. However, the physics of fluid flow is providing important clues. The basic idea is that fluid does not move at the same speed throughout a tube. Flow velocity varies from one location to another. Near the walls of a vein, the cytoplasm flows more slowly than it does in the center. Why? Because friction slows it down. When friction within a vein becomes relatively strong, the vein tends to shrink. When friction is weaker, the vein tends to grow. Strong shear forces therefore promote vein growth, while weak shear forces contribute to vein degeneration. However, this explanation alone cannot fully account for what researchers observe. Another factor appears to play an important role: the architecture of the network itself. The overall structure of the network influences every individual vein, whether large or small. For example, if a small vein is located next to a particularly large one, it is more likely to disappear. If that same vein is surrounded by other veins of similar size, it is more likely to persist. Importantly, both degeneration and the state of a vein remaining where it is occur with a slight delay. That is because cells require several minutes before they can respond to changing conditions. And this delay presents a major challenge for researchers. It makes it more difficult to determine exactly how the entire network influences each individual vein, and at present, this is where our scientific understanding reaches its limits. But why should anyone care about why a vein grows, forms, or disappears? Who could possibly benefit from this knowledge? The answer is quite simple. Slime molds effectively program their own vascular networks. If we can understand how they accomplish this, we may one day be able to program vascular networks ourselves. In other words, we could potentially influence the growth of our own blood vessels. We might learn how to control vascular remodeling. We might be able to cause newly formed blood vessels that supply nutrients to a malignant tumor to regress. We could potentially develop more effective treatments for diseases involving abnormal changes in arteries and veins. The list of possibilities is endless. As a result, researchers are actively studying these processes and simulating blood capillary systems using biochip models. Slime molds have so much more to offer than most people realize, and there is still a great deal that we do not know. Research on Myxomycetes is not driven by curiosity alone, since there are also potential benefits for humanity. Research in this area could prove itself to be incredibly rewarding. And with that, we have reached the end of my podcast about slime molds. Give me a little more time, you will certainly be hearing from me again. I hope I have sparked your curiosity about Myxomycetes, and thank you for tuning in today. Goodbye.
Sie haben gehört, auf den Spuren der Schleimpilze. Schön, dass Sie heute dabei waren. Bleiben Sie dran.